Hello everyone, today we are going to discuss the chapter regarding nutrition. In this chapter we are going to discuss detail regarding what is nutrition, what is the beneficial of nutrition to all living organisms from single cell organisms to multicellular organisms. We will discuss detailedly in this chapter. So first, nutrition. Nutrition is the process by which organisms obtain food and utilize it for growth, energy, repair and maintenance of the body. Whatever the food that we are taking, that food is used for growth, for body growth, then energy. With help of energy, I am able to speaking, I am able to writing. So we need energy, then repair of our body and maintenance of our body. Repair and maintenance. Simply whatever the food that we are eating, that food is used for body growth, energy, repair and maintenance of our body is known as nutrition. So this nutrition is classified into two categories. One is autotrophic nutrition and the second one is heterotrophic nutrition. Nutrition is classified in two categories. One is autotrophic nutrition, second one is heterotrophic nutrition. So we'll see detail regarding what is autotrophic nutrition and what is heterotrophic nutrition. First, coming to the autotrophic nutrition. Autotrophic nutrition means it's a type of nutrition the plants will prepare their own food is known as autotrophic nutrition. For example, autotrophic nutrition. Simply auto means self. You have to know auto means self. Of nutrition will be prepared in the presence of photosynthesis. All green plants are able to prepare their own food is known as autotrophic nutrition. Then coming to the heterotrophic nutrition. Heterotrophic nutrition means the organisms which cannot prepare their own food but depend on other organisms for food is known as heterotrophic nutrition. Simply these organisms will not depend food by their own but depend on other organisms for food is known as heterotrophic nutrition. This heterotrophic nutrition is classified into four categories, that is holozoic nutrition, saprophytic nutrition, parasitic nutrition and symbiotic. So we'll discuss one by one what is holozoic nutrition, what is saprophytic nutrition, what is parasitic and symbiotic. So as I said you, autotrophic nutrition, organisms prepare their own food. So simply whatever the organisms that will prepare their own food is known as autotrophic nutrition. The best example is green plant. All green plants will prepare their own food in the presence of photosynthesis. Then heterotrophic nutrition. Organisms depend on other organisms. Simply these organisms cannot prepare their own food, so they have to depend on other organisms for survival. So these heterotrophic nutrition were divided into four categories. Holozoic nutrition, example is human beings and amoeba. Holozoic means simply whatever the food that we are eating, the food is digested inside of our body is known as holozoic nutrition. After taking the food, the process of digestion will occur inside of our body is known as holozoic nutrition. Examples are human beings and amoeba. Then coming to the saprophytic nutrition, example is given as fungi. Saprophytic nutrition means the organisms that which will feed on dead and decaying matter is known as saprophytic nutrition. Simply these organisms will depend on dead and decaying matter is known as saprophytic nutrition. Example fungi. Parasitic nutrition, it's a type of nutrition the plant will attach it to another plant and they will get food directly from the host plant body is known as parasitic nutrition. Example cuscuta plant. Then symbiotic nutrition. In this type of nutrition, both organisms are getting mutual beneficial to each other. In the symbiotic nutrition, both organisms were getting beneficial by sharing each other. It is known as symbiotic nutrition. Example lichene given. So coming to our chapter nutrition, so we seen regarding what is types of nutrition, that is autotrophic nutrition as well as heterotrophic nutrition. So we seen autotrophic nutrition, heterotrophic nutrition. Then coming to photosynthesis, all green plants will prepare their own food by the process known as photosynthesis. For the preparation of photosynthesis, the plant should get availability of sunlight, proper amount of carbon dioxide and water from the soil, ground itself, and chlorophyll. The chlorophyll pigment will present inside the green leaves. Along this, if the plant should prepare therefore there is a necessary for water, the water should be available and chlorophyll. Then carbon dioxide and sunlight. In the presence of these four categories, then only the plants are able to prepare their food. In the presence of water, chlorophyll, carbon dioxide and sunlight. Without presence of these four elements, there is no formation of preparation of food. Then coming to here, so in the presence of carbon dioxide and CO2 to H2O, in the presence of light and chlorophyll, this reaction is going to occur in the presence of light and chlorophyll. The end products are observation is seen the sulfur bacteria. C.B. Van Niel was a scientist. He observed regarding the purple sulfur bacteria. He found that this sulfur bacteria able to prepare their own food by using the hydrogen and sulfur. Then he given the equation. So this is the equation for photosynthesis. In the presence of carbon dioxide, water, in the 6CO2 plus 12H2O, in the presence of light and chlorophyll. Whatever the reaction, that reaction should happen under the presence of light and chlorophyll. Then the end products were C6H12O6 plus 6H2O plus 6CO2. These were the byproducts, end products of the photosynthesis. So now next coming to air and photosynthesis, we'll see with an experiment. That is Priestley experiment, air and photosynthesis with help of an Priestley experiment. So in this experiment, the metal requirements were, bell jar, these are bell jar, candle, mouse, mint plant. These were the metal requirements. So coming to the process, what we have to do, in the first situation, the first case, we have to place candle in the bell jar. So after placing the candle, burning candle, after 5 to 6 seconds, the candle will turn off. Then the second situation, he place burning candle as well as mouse. After some time, what happened? The mouse will get difficult in breathing and the candle will turn off. In the third situation, he placed mint plant, candle as well as mouse in a same bell jar. So the candle is able to burning as well as the mouse is able to living properly and plant is also able to surviving properly. So from this experiment, he said that whatever the plants is releasing oxygen, that oxygen is utilized by the candle as well as the mouse. Whatever the oxygen is released by the plant, that oxygen is utilized by the candle as well as mouse. Then this mouse is able to releasing carbon dioxide. Whatever the carbon dioxide that is released by the mouse, this carbon dioxide is utilized by the plant. So by this exchanging of gases, these three, the mouse as well as candle able to survive without getting any suffocation. Clear? Then coming to next topic, that is activity 2 regarding the carbon dioxide is necessary for photosynthesis. For preparation for the formation of photosynthesis, carbon dioxide is also necessary. With help of an experiment, we'll do this. We'll prove that carbon dioxide is also necessary for the formation of photosynthesis. So coming to the material required, these were the material required for the formation of carbon dioxide. What are the material required? You have to take the potter plant. Then you have to take a transpirated white mouthed bottle which is transpirated KOH, potassium hydroxide and split cork. Then you have to take the iodine solution also. So procedure, what are the procedure that we have to do? First you have to take the potter plant and let it destarched. What is mean by destarched? Destarch means removal of starch present in leaves is known as destarch. For doing the discharge, you have to place this potted plant for 2 to 3 days in a dark room. In a dark room for 2 to 3 days, then only the plants will get discharged, means completely removal of starch from the leaves. So you have to take the discharged plant. After that, after taking discharged plant, you have to take the white mounted bottle as shown in the figure. Inside the white mounted bottle, you have to add KOH, potassium hydroxide solution, in the white mounted bottle as shown in the figure. Then after adding that one, you have to place, after adding potassium hydroxide, you have to place half part of the leaf inside the bottle. You have to place carefully half portion of the leaf covered inside the bottle and half portion of the leaf outside of the bottle. Then you have to cover the bottle with the splitted cork carefully. So place it under the sun for 3 to 4 hours. Simply after arranging this experiment, I am repeating once again, you have to take the discharged plant. After discharged plant, you have to place the half leaf inside the white mounted bottle. Before that, you have to place potassium hydroxide solution inside the white mounted bottle. Then after arranging this experiment, close that white mounted bottle tightly with help of the splitted cork. This splitted cork will prevent the entry of the atmosphere gases to inside the bottle, as well as escaping the gas particles from inside the bottle to outside the environment. Then after this, you have to place this arrangement under the sun for 3 to 4 hours. 3 to 4 hours you have to place under the sun. Then after placing under the sun, you have to, what happens, you have to know this potassium hydroxide. Whatever the potassium hydroxide present inside the white mounted bottle, this KOH, potassium hydroxide, will absorb the carbon dioxide present in the leaf. Whatever the carbon dioxide present inside the leaf, it is able to attracted, absorbed by the potassium hydroxide solution. For that reason, we are placing the potassium hydroxide solution inside the white mounted bottle. Then after that, you have to plug that leaf and place it in a petri dish. Then you have to do the iodine test. Iodine test, whichever the plant, the pot plant which is exposed to the sunlight, which is outside of the bottle. This is outside part of the bottle and this is inside part of the bottle. So after doing the iodine test, this pot is able to forming into bluish black color. So bluish black color is an indication for the presence of starch. Whenever the leaf is able to forming into bluish black color, then that is an indication for presence of starch. The colorless will indicate there is no starch. Clear? So by this experiment, we are able to say that carbon dioxide is must and should for the preparation of photosynthesis. Simply whatever the leaf which is present inside the bottle, the carbon dioxide particle was absorbed by the KOH. There is no carbon dioxide was present, but we kept in under the sunlight. The sunlight is able to coming, but absence of carbon dioxide, the starch was not formed. Clear? So simply by this experiment, we can say that carbon dioxide is necessary for photosynthesis. Clear everyone? Then coming to next, and one more thing, this experiment we call as moles of experiment. Then coming to lab activity, experiment to observe oxygen is evolved during photosynthesis in the presence of light. So what is saying means, during the photosynthesis process, green plants will release oxygen. So we can prove with an experiment. We can prove that during the photosynthesis process, the plants will release some amount of oxygen. So for this experiment, the metal requirements were, what are the metal requirements? The metal requirements were hydrilla plant, funnel. This is a funnel which is transparent. Then this is a test tube, water, beaker. These were the metal requirements. Procedure. First, you have to arrange the apparatus as shown in the figure. You have to take the beaker, fill the beaker with water. Then you have to take the hydrilla plant. That hydrilla plant should be placed inside the transparent funnel as shown in the figure. Then you have to submerge the funnel under the water at the base of the beaker. Then after that you have to place inverted manner test tube. Okay, you have to place the test tube on the above of the funnel. Then place this setup for 4 to 5 hours under the sun. After arranging this apparatus as shown in the figure, then you have to place this arrangement under the sun for 4 to 5 hours. Why I am placing under the sun? The plants will prepare photosynthesis in the presence of sun. For that reason, you have to place it for under the sun for 3 to 4 hours or 5 hours. After that one, carefully you are able to observe that there is a formation of bubbles and there was a space was filled here. So why these bubbles, why this space was filled means during the photosynthesis, when you place under the sun for 3 to 4 hours, the plant will prepare photosynthesis in the presence of sunlight. So during the photosynthesis, the plant, whatever the hydrilla plant, this plant is able to releasing some amount of bubbles into this funnel. This bubbles is known as oxygen, simply oxygen. During the photosynthesis, during the photosynthesis process, we are able to see clearly some amount of bubbles are evolving. These bubbles are collected by the test tube. So these bubbles are known as oxygen. By this experiment, we can say that during the photosynthesis, the plants will prepare the oxygen. So after this, you have to take out this test tube clearly without escaping of this oxygen. Then you have to place incense stick inside this test tube. You have to place the incense stick or matchstick. Then what you see, you are able to see that the matchstick starts burning rapidly. So by this we are able to say that oxygen helps in burning. So from this we are able to say that simply oxygen is released during the photosynthesis process. Then coming to next experiment. Activity 3 regarding sunlight is necessary to form starch in green plant. So whatever the starch that is presenting in green plant, the starch is able to prepare under the presence of sunlight. Simply, if the plant want to prepare the starch, means if the plant want to prepare the food, that food is able to prepare under the sunlight. So with help of an experiment, we can see this one. So first what you can take a potted plant with a de-starched leaves. So in this experiment also we have to take the potted plant with de-starched leaves. Simply the starch, whatever the starch materials present in leaves, we have to remove. So we know that while you're placing this plant under the dark room for 3 to 4 days, the plants will get rid of completely starch. Clear? So if you want to discharge this plant, you have to place under a dark room for 3 to 4 days. Then after 3 to 4 days, the plants will get discharged. Then first what you can take a potted plant with discharged plant. Then remember the process of discharging leaves mentioned in the above activity. Simply we have to place under dark room, then only we can get the discharged plant. In the second step, what you can cover one of its leaf with a black paper which is design card. Simply you have to select any one leaf. After selecting, you have to cover half portion of the leaf. You have to cover only half portion of the leaf with a black strip, and you can make any letters as you want. Then what is given? Place this potted plant under the sunlight. After arranging this black strip, you have to place it under the sunlight for preparation of photosynthesis. Whenever the sunlight, photosynthesis will take place. Then what again? After few hours, exposure to bright sunlight, test the leaf which is covered by black paper for the presence of starch. So after placing under sunlight for 3 to 4 hours, you have to pluck this leaf and check this leaf whether starch is prepared or not. So by the observation, the result will be the cover strip which is covered by the black strip is colorless solution. By adding the iodine solution is very colorless. The leaf which is exposed to sunlight is turned into bluish black color. Simply the leaf which is covered by the black paper will turns into colorless. Simply colorless means no starch was prepared. The leaf which is exposed to the sunlight, the leaf was turns into bluish black color, blue black color. So whenever the leaf is turning into bluish black color by the iodine test, there is an indication for presence of starch. Simply by this experiment we can prove that starch is prepared under the sunlight. Clear?